been practicing with my drone, and I gotta, at least with that thing, I gotta fly over at least 6 different angles to get anything decent.
Can we zoom that one? I think it's different.
Sure. Okay, I got a tow out there. You should be able to full zoom there. Get some polyps maybe.
Oh, nice shot. All right, thank you very much.
Okay. You can go wide, thanks. Looks pretty steep.
Yeah, it's, I mean,
That's where we gotta go.
we were planning the dive. It was, the average but the drop was going to be 20 degrees or so.
I'll wait till Atalanta gets out there and drop Atalanta down behind us.
All right. Yeah, I'm going to keep the steps coming. Keep things moving, yeah?
Right there, yeah.
Bridge nav, 40 meters, 165.
Might be an interesting still camera with all black in the back. I could get it a little closer maybe.
There's a mullet. All right, I think we're good back here.
Right. We're gonna spin Atalanta around and look northish, or maybe I'll come off the side of you. Not sure which side yet. Just so I could stay looking uphill. Shrimp is arching its back.
Yep.
You want to spin Atalanta around, Jade?
Yep. Which way?
You can come clockwise there and look at her.
Looks like the sun has snuck up when we weren't paying attention in the dark, cold control van.
What time does it come up? It's like
5 something.
5 something.
I think it comes up and sets at about 6 o'clock.
Hmm.
that make our antenna happy if we do that? Yeah. You'll have to come right down now. You're deeper than I am. You got enough leash to look to your right now?
Let me check.
Another one of these black
Nope.
corals we looked at earlier.
Start going a little. There we go.
You can come down a few more meters if you want.
Are we still making our way down?
Yep.
Oh, okay.
We will be for 30 minutes or so probably. Maybe a little bit longer.
No, you want to stop it up there, Chris, till I catch up.
Yep. Bridge nav, hold position.
Any of these rocks might be loose?
Yeah, could be. I can get down in there. I think we can poke a few there. You get the minute out if you want, Jaden, and poke some of those rocks. You never know.
Yeah, it's
should be
annoying. They look so loose from
It
2
should
or
be
3
hot.
meters, and you get down here and they look pretty cemented.
That rock looks interesting.
Yeah,
Is it a
that-
coconut?
Yeah, there's a coconut in
Oh,
there.
never mind.
Coconut.
Do you guys ever find like different organisms living in the coconuts?
I've only ever brought up a
You'll have
coconut
to
once,
swing left in
and it
there
had
and try to
very
bounce
little
the rock.
living on it. It might have just not been down there
Otherwise-
very long or whatever, but it was not particularly interesting.
Not like the wood, huh?
Not like the bigger pieces of wood we've been seeing. I have no explanation for that. Like I said, it's probably just the coconut I happened to bring up was just fresh, but
It would be interesting to study the different communities on different types of plant detritus.
that one's loose.
Is that too big?
Rounder one would be better.
Yeah, the flat ones are crust.
That one maybe.
I think it moved. Yep.
How about this one?
Yep.
Can we pick that one up and see how thick it actually is?
Yep.
No, too thin. That's just friable material. What about this one?
Nope.
Nope. That's gotta be too thin, I think. And too big. They look like there might be some more, this little promontory just on your right ends. Maybe we can hop back up 2 meters or something and look at the base of it.
Yeah, You can just tuck that in over the porch and I'll hop back. That's good there, you can hold that.
Copy.
Come on, Herc. Okay, we can poke around in there and find something maybe. Just to your right is, I think, what Brian was on about.
Any specific ones you want me to poke?
These all look very thin. I don't think we're gonna find a good angular, thicker rock here. This all looks like crunch crust.
Okay, hold what you got there. I gotta come up here.
Copy.
You can park it. Just gonna slide to the south a little here.
That's our first prim note, I think, of the dive.
Can you tuck it in? Oh no, that's fine there. Never mind. You're good there. Good
Okay.
there. Good there. I was thinking we'd find another one, but
So you guys were able to test some of these rocks here in the lab?
Yeah, we acided a couple of them, and they pretty much are all coming up as
it should be
as
enough
carbonates.
slack to spin around now.
The geology here is really blowing my little biologist brain. I do not understand how much carbonate we're seeing in places, and at the depths we're seeing it, which is good evidence for Chris's argument that this is carbonate too. Which is consistently surprising me, how deep we're seeing the carbonates here and how old these seamounts seem to be compared to what I would have expected. I thought they would have been mainly back island arc or back arc volcanism from the trenches, and they seem a lot older than that to me. But again, the biologist thinking, is it playing geologist on television?
It should be good to come up a little bit there as I come under you.
Copy.
Big Anthomastus over there. Big stalked crinoid.
And look down a little for me.
Copy.
Perfect, thanks.
Does that rock want to come home with us?
There could be some little cobbles in there.
Nope. Never mind. I can tell now. That's all too flat to be. Give us a good rock sample. Never mind.
Heard it. I was just going to say, I think we're good for 20, Chris.
So what dimensions are you looking for?
Now the trick is with the carbonate is it forms a rind around it, or it can form kind of a planar thing, and so we're looking for something that's roundish and ideally has more angular lines. Makes it more likely that there's a core rock, there's a core native rock in there that's big enough for us to actually look at, as opposed to it just being all all manganese crust.
There's the first Iridogorgia.
Yep.
The dive.
As the geologists call them, firecracker corals.
Sorry?
Or fireworks coral.
Oh, yes. There's another one.
Yeah, look down a little more, Jane.
Yep.
See if there's any big obstacles before I hit them. Sorry, before I slowly, gently bump them. I'm watching on my rail cam. It's pretty benign there. It's those big
Nice little
whips
patch
and
of
stuff I can't see.
Paramaricid sitting here on this plane. Some more Victorgorgia, Calyptrophora, a couple of Victorgorgias. Definitely seems to be a little more sediment load on this side than when coming down the feature than the northeast where we came up it.
Was something munching on this one there that
It's kind of interesting, the crinoid right there,
Yeah.
right where the tissue starts. I wonder if there was a sea star making its way up and then stopped at the crinoid.
There's also something white there that might be a snail. I can't tell what
pushing there just a little?
that
first?
is. Yep, that's a snail. So that's what's eating the coral there.
That little snail?
Yep.
You think so?
I don't disagree. That seems like a very tiny snail to have eaten that much coral, but there definitely are some snails that will eat coral. And given that the snail is hanging out right at the interface between, I don't know, it's possible.
Could be that a larger sea star stripped away the coral tissue and then there's some kind of biofilm that grows on the dead skeleton that
Yep.
the snails eat.
That is also 100% possible.
Okay, okay, go ahead.
Hey, folks. Does anyone need anything from the data side of things?
Not at the moment.
Yeah, and the data lab is a cold Starbucks in the fridge.
All right, I gotcha.
I love you forever if you bring me one.
Looks like there's Metallogorgia hiding in there as well as the Paramyrcids and the Victorgorgias.
Yep.
So good morning from Palau. It is 6:26 on a Wednesday morning here. We are on a 23-hour dive today.
What's that? Yeah, yeah, all good. All right. Looks like you got a little local uphill behind you there, Jaden. Something to look at, maybe.
Oh, whatever caused this terrain is really cool.
Yeah. The verticality of these walls makes
Yep.
me lean towards fast cooling lava tube type formation, but
I give up. I want a geologist.
yeah, speculating.
Yeah, I wonder if it was like slow moving lava or...
And now Chris is saying this is additional carbonate evidence. I don't understand.
Does it make you want to go back to school for geology, sort of?
I have a bachelor's in earth science.
Oh.
And I don't know. I don't know what's going on.
No, I just want to phone a friend geology. But it's very cool nonetheless.
It looks similar to axial, where there's a thin crust everywhere with lots of pillars and stuff underneath it where it's fallen away. You come down a bit for me, I can get around here.
Copy.
I don't quite have that. And none of this stuff was in the big map.
Nope. Any of these things down here look, rubble piles look sampleable.
Have a look. Thank you.
Chris, getting involved. Yeah.
I'm gonna do an opportunistic zoom on the
Yep. Check out the sea cucumber.
Nope.
Have you seen any other geology formations like this before, Brian?
This is different. I've never seen one quite like this.
That's amazing.
I mean, on this cruise, there have been some like this.
Similar, but not. I never seen like so many pockets.
Look down just a bit for us, Jane.
Copy.
Good shot of her clear.
All the more important to find a rock sample, I guess.
Yeah, I have to admit, I'm becoming more and more convinced it looks like carbonate and
Yeah,
karst
I'm
terrain
also
down here.
starting to switch sides.
But I still really struggle to understand the larger regional plate tectonics and subsidence pattern that explains carbonate down here at this level. You don't think anything in there that's gettable looks like it's going to be a good rock?
Just gonna change the angle of the dangle here.
All right, I'm going to go below and take a nap. I'll be back up for breakfast.
Roger. Thanks, Rachel.
No problem, thank you.
Push in for me, video. Actually, sorry. You can go a little wider. There's one right. No chance.
Oh yeah, can we take that one?
Sure.
Just kidding. That has potential though.
Zoom in there for me, please. So it's pretty crusty.
It's pretty crusty. Let's leave that one. That one's crusty too. Leave that one.
Yeah, I'm just gonna pick it up, see what rolls out there. Maybe a potato under there.
What about this one under the arm? Here.
I poked that one.
You did? Okay.
I couldn't get it to move. A butterfly rock.
Yeah, that one's so spread out. That's definitely crust
But it's crust with,
with potentially something on it.
yeah,
All right.
I don't know. It'll fit in one of the
that's
big boxes.
yeah, that's the best thing we've gotten so far. So
It had the potato shape on top, didn't
yeah.
it?
Well, let's take that one. It's the best option we've gotten so far.
Right. I'm gonna have to come up out of here. I'm in a bit of a hole. Stay. Can zoom tight on that rock for a minute while we're coming up if you want. Okay. You want to open the starboard box for me and put the rock in there?
Copy.
Give it a little. Wash machine action out there. Okay. Now I'm ready for the move.
Sample number?
What's that?
83.
Thank you.
Can you find Herc in that?
Yeah.
Someday I'm going to put the other 180 cam on there, but maybe next cruise.
I find it interesting that we rarely see lots of different groups of stalked crinoids together. Generally, it's one, maybe two types that we see, and then see that one or two types over and over and over again.
Yeah, they're not super diverse, it seems.
Yeah, but I mean, even amongst the what is diverse, it's, I know, dive by dive and different depths, they seem to be pretty specialized.
Yeah, I don't know.
Like we'll go through a dive and we'll see basically one type for most of the entire dive. Sometimes it switches over, but a lot of times it stays pretty consistent. We almost never see a field of 3 or 4 or 5 different types of crinoids, stalked crinoids together.
I feel like I've been here before.
Seeing all these holes in the rock into the lower things makes me think of a dive on the lee side of Lanai called what's it called, Cathedrals, I think, where it's a big space like that that you can actually scuba dive through.
Cool. And is it carbonate or basalt?
I think it's carbonate. I'm amazed how much we're going up here, contrary to the large-scale bathymetry. Oh, and here's the second type of crinoid, just as I open my mouth. The second type of stalked crinoid.
Sorry, just getting a sense of what's
Yep.
out there first.
No problem.
Yep.
Good for another move, Chris.
So the stalked crinoid we're seeing now, according to Chris Kelly, is, I'll try and pronounce this, Iselocrinidae. Another purple Victigorgia. Oh, sorry, that was referring to the previous one. The previous stalked crinoid with the little leg appendages. This one is Pheronocrinidae. Whoa. Is that a piece of wood? Let's take a look at this.
All right.
Wow. Yeah, it's another big piece of wood. Looks like it's come down and landed on some of these corals. Let's zoom on the shrimp side. And then maybe come along from right to left.
Okay, can push in a bit there for us.
Very interesting. There's another little squat lobster. And if we could just slowly scan along the branch,
Yeah, go ahead.
that'd be awesome.
I'll try and hold it here for you.
Copy.
Yeah, these munidopsid squat lobsters. Been seeing them on a lot of the woodfalls around here. And there has been quite a lot of woodfalls throughout the entire cruise. Woodfalls can host specialized communities of animals with chemosynthetic symbiotes, similar to whale falls, but with a different set of microbes degrading the wood itself and producing chemical byproducts that are used in various metabolic microbial pathways that animals can take advantage of.
Maybe tilt down a little. See.
Now we can see this log is full of life.
No more fin.
I can rotate my head a little bit.
And a little sea urchin there at the end. And yet another unidopsid. Oh, I didn't even see this chrysogorgid. That's great. Are we at max zoom right now? Perfect. I'd just like to see some of the stuff coming off the end over here. Some reasonable worms.
Can you focus on the wood in the background there? Perfect, thanks.
Thank you. And another squat lobster. And then if we could take a quick look at the chrysogorgid, that would also be great.
You want to tilt down
I
a
don't
little,
think we've
Jake?
seen this one yet.
Yep.
On this dive. Thankfully it was spared by this log rolling downhill.
I love their beady little eyes.
Is this a Europtychus?
I believe so, yes.
All right, I think we're happy. Thank you.
Okay, you can go wide for me.
Yeah, it's interesting how there's a coral just uphill of it that is not bent over. So I wonder if the log just landed like that, or the coral had some flexibility, or...
Okay, Chris, I think we're ready.
Oh, that I did not know. Chris is sharing fun facts about crinoids.
Yeah, I didn't know that either. Chris is saying that many, if not all, unstocked crinoids begin life as stocked crinoids. They reach a certain size and then break off the stock. I did not know that. It's very cool. They break off from part of their stock, leaving the upper part still attached.
There's a Victorgorgia about to come into view. Can we take a quick look at it when we get there?
Yeah. Can you speed up the tilt down just a little, dude?
Yep.
ourselves up. Alright, should be able to push in on the purple one there. Perfect, Jaden, thank you.
All right, thank you.
Okay. This control van needs some bobcat chairs in it.
Ouch.
I thought I would not like it at all until I spent 12 hours in one. Like the footrest that lays up and you're almost vertical while you're, they're almost horizontal.
Are we moving up or?
We're still moving down.
Oh, okay. ROV pilots, is it hard to go downward or
No.
challenging?
It's, yeah, it's not as easy as looking at a vertical wall, that's for sure. It's challenging to get good video for the scientists so they can actually have something useful to, because they're still annotating all this, right?
Yep.
Still covering ground and still burning bottom time.
Yeah, just in its simplest form, the camera is just further away from the seafloor. Or if you're coming down facing away, or you're backing up and the pilot's having to fly, you know, through situational cams or looking at Atlanta. So it is just slower and not as good a video.
Yeah, it is definitely slower. Like, we could go a lot faster, but then I stir up all the, you know, because we're going backwards, looking at my dust cloud. So I try to, depending on the terrain, we zigzag or go sideways. Yep,
Yeah,
you can keep moving.
whether or not to come down this feature was the primary debate at the dive planning meeting at 2 a.m. this morning, was whether it was worth the time to see this feature that it would take to come down backwards.
Yeah, totally worth it.
That was clearly what we agreed upon.
You have to come down, Jaden. End of my leash there.
Another Ritagorgia hiding in there.
I'll come back to your right for a while so you can look into the hill.
Do corals and sponges have like any control on where they land or grow?
Yes,
They
they have a mobile larval phase. What controls that in the deep sea is very much unknown. What controls that in shallow water is an area of very hot research, and it's way more complicated than you expect. You know, there's chemical sensors, there's in the water, there's chemical and texture sensing on, you know, the seafloor. There's strong evidence that even sound and the noise of a healthy reef is more likely to attract larvae. So there's a lot of different cues that it looks like corals can use in locating settlements, and there's almost no work on that in the deep sea yet.
Yeah, it just seems like the ocean's just so complex that there's just, because there's so many variables, and yeah. Does it feel sometimes like a little overwhelming?
Unquestionably, yeah.
Yeah.
Especially when you think about all the things that we know might play a role and how complicated and different they are to measure. Couple that with having to do everything through an ROV or a submersible, that trying to ask these questions in the deep sea definitely sometimes feels daunting.
Can you start looking to your right some, Jed? Try and look into the hill there.
Can we look at the crinoid, please?
Yeah, right there. Sorry, I'm looking at the sonar. This one here?
I think so. Chris, is this the one you're interested in? Or did we miss the one you were interested in?
Push in there.
Yep. Can we get a really tight shot on this one, please?
Yeah.
We'd like to look at the stalk side of it.
Roger.
Oh, that's a Tudoscarium that's hit the sediment and is caught up in its arms. It's a very large radiolarian. So
Wow.
this crinoid's
Cool.
stalk is substantially shorter than
Yeah.
most
See how it's
others.
grabbing on with its little legs. Cool.
You're going to have to do the camera for me, Jed.
Copy.
Chris, do you think this
Tilt
thing
up a bit
got
so
its head bitten off and
you can zoom in.
regrew with a shorter stalk, or?
Tilt up, Jed.
Copy.
This is one of the stalked crinoids that walks. Some are permanently attached, but this is one of the stalked crinoids that can actually walk around,
Okay,
just like
good zoom in.
unstalked
Video.
crinoids.
That's fascinating. I think I'm gonna Google that later too, or YouTube it.
I was
Is there
saying
footage
tilt
of
up
that?
on the HD
There is.
camera. Sorry.
There is.
Oh,
It looks
sorry.
very strange.
You can go maybe a little bit tighter there.
All right, Chris, is this
Roger.
what you got? What you need on this one? All right, we're good. Thanks.
Okay. I can go wide, thank you.
Crinoids are a
back
quintessential
down now.
example of a living fossil. So something that was previously only
I
known
want it.
from the geologic record and then was later discovered in the deep sea. It was a pretty pivotal
You have
early
to, tilt
scientific
your camera down a bit for me.
advancement in deep sea exploration. Led a lot of people to think that maybe the deep sea was a
Sorry,
reservoir
fire
of
maintenance while we're going
prehistoric
backwards.
species. But it turned out we just needed to do some more exploring.
Yeah.
I have a question.
Mm-hmm.
How closely are these related to ferns and other land shrubs?
So despite the fact that they look quite a lot like plants, they are animals, and they're more closely related to sea stars and sea cucumbers than they are to plants.
Oh, interesting.
So
Thank you.
there's really, the only plants we're seeing down here are these logs and
coconuts.
coconuts raining down from the surface. So there's no light for photosynthesis, can't support single-celled phytoplankton.
Yep.
but the sort of plant-like body form serves a sort of analogous purpose, which is to increase its surface area and catch particles in the water column, which is sort of like how plants' leaves increase their surface area to capture sunlight. So they're doing sort of an analogous thing in the ecosystem.
Well, thank you for that.
Mm-hmm.
So this is another bamboo coral.
So that's interesting you say that, Harold. So there would or would not, or would there be fungus at the bottom?
Yeah, so there is actually fungus in the deep sea.
Okay.
So surprisingly, fungi, the kingdom fungi, is actually more closely related to animals than they are to plants. So just like us, fungi breathe in oxygen and exhale carbon dioxide, whereas plants do the opposite.
Okay.
And there are single-celled fungi all over the world and even in the ocean and even in the deep sea. And this is a really interesting emerging area of research. Fungi are really interesting because they produce lots of chemical compounds that are really interesting to researchers who study pharmaceuticals and natural products. They're biochemically very, very interesting and very different. They also might play some roles in biogeochemical cycling that we haven't totally figured out yet. And in shallower waters, there's also research about how they're related to diseases. So yeah, deep sea fungi. Who knew?
Yeah. Thanks.
Mm-hmm.
So you are watching nautiluslive.org. This is the second voyage of Libu's voyage, part 2. We have been on the ship. This is an 11-day expedition.
And this is our last dive of this expedition.
Yeah.
We're going to be in the water for a good little while, probably another 20 hours or so. And then we'll start our transit back to Core.
All right.
So we're getting down towards the bottom of the slope where we're probably going to find some sediment and run away a little bit of a saddle in between this local little peak we are been exploring and then getting on to a much larger ridge that'll take us almost 700 meters shallower as we traverse it throughout the day. Can we go look at the big coral, please?
Absolutely.
Yeah, Chris, this is where we started the dive, is looking more like this on the other side at this depth, which is, I think, why Harold and I were biased towards our earlier opinion when we saw the transition.
Push in a bit there for me, please.
So this is a primnoid, probably a Cliptrophora. It's got an anemone living on it. And it looks like we've got a cup coral and another anemone right next to its base.
It's got some interesting secondary branching patterns going on.
Yep. Can we kind of pan around and see if it's got any other associates living on it?
Sure. Go, you can push in a bit more there. Go ahead and push right in. Is that all full zoom there, is it?
All right, I think that's probably good. We can continue on, thank you.
All right.
So we are in the middle of waypoint 1 and waypoint 2, right? Like right in the center?
Pretty
sort
much.
of? Yeah,
Yeah.
halfway.
Okay, can go wide for me, thanks. Hey, I can see the other side Atlanta sonar now.
And here comes a fish. Can see it in the psychic camera.
Oh,
Oh, yeah.
looks like a rat tail. Ooh, yes. Please zoom here.
Yeah, go ahead and push in there.
This rat tail looks like it's seen better days.
Yeah. Well, just yesterday, Jeff and I were remarking how we hadn't seen a lot of these. This looks like a Corphinoides rat tail. These larger rat tails that were very common in the beta camera study. And this one looks just like one of those.
Okay, I
Alright.
can go wide.
Sounds good, thank you.
Looks good.
Couple more Calyptrophora. Another urchin, like we've been seeing a fair number of. That was a stark transition. Come off that rubbly pillow basalty thing into this kind of flat terrain now.
Roger. Roger. Good to keep moving.
This looks different. Let me look at that whip, please.
All right.
Oh yeah, what is that? Big sea pen?
That's my initial
Yes.
Interesting.
We shall know here in a second. Yep. Think that's a C-pin.
Wow. It's got to be one of the bigger sea pens I've ever seen. Halopterus. Thanks, Chris.
Hey, that's good with press,
Uh,
thanks.
You want a close-up?
Oh, actually, never mind.
Yeah.
Can we land and do a super close-up on the polyps?
Yeah, you can actually push in there or get a good shot of them. Go ahead, go full zoom.
So we're looking for pink spines.
I think I saw some, but we'll see here. Yeah, these. Are these the spines?
Looks like spines to me.
It's a bit bouncy. I can land if you want a super steady view.
Looks like this has also been damaged. Almost looks like that's been kind of partially degloved, where the tissue got torn and then pulled down the end of the stalk and is kind of sloughed off the top.
All right, I
All
think
right,
we
thank
got
you.
what we need. Thank you.
I'm still just blown away by the giant heteropod from yesterday's dive. That thing was amazing. Never seen one of those before. Did you see that video?
I don't think I saw it, no.
You have to go look at this video.
A heteropod?
Heteropod.
Wow.
It was crazy.
That's weird.
Yeah, I haven't seen it either. I heard about it from an ROV pilot description. So.
This is a common question that keeps occurring, but could you briefly just explain like how, like if we do find species that we don't know, like the naming process and how that works?
Yeah, so the naming process for a new species actually takes quite a long time. So first we have to actually collect a sample of it. Then that sample goes to a museum where it might sit on a shelf for quite a long time before it gets the attention that it needs to be described. And typically that'll be done by a taxonomic expert who takes a look at the species and measures a whole bunch of things about its morphology. And nowadays it's much more common to do genetic analysis of it, to include that in genetic libraries. And then it's really up to the person who describes the species to give it a name. And there's sort of a movement now towards selecting names that honor the places that the samples and species were collected from, that have a connection to the places from which they originated, and sort of a move away from just naming species after
I'll come back towards you.
random scientists
Oh yeah.
or other things. But yeah, the naming process is up to the person who describes the species, since that is the bulk of the work that goes into that.
And it's a pretty laborious and underappreciated work too. It takes a lot of work, and there's actually fairly little credit given scientifically in academia to the people who do that kind of work.
Thanks for that.
Yeah, there's actually one of the sort of scary things happening right now in the field of ecology is the sort of decline in the number of new people studying taxonomy. So a lot of the world's experts on various animal groups are, getting older, retiring, without necessarily new scientists coming in to fill in those gaps in expertise. So if you're out there watching at home.
And if you're a funding organization, you have to hire taxonomists too.
Exactly. And
there is a shortage of young taxonomists, but I also know several young aspiring taxonomists who are struggling to find taxonomy jobs. So it's not just the lack of the people, but it's also the lack of, that's a cool Walteria. So this is a Walteria sponge. It's got kind of a twisty growth form, which is a little unusual with an anemone sitting on top of it. Look how many brittle stars are on it too.
Wow.
All right, thank you.
Okay.
Yeah, is it twisting or is it bulged out at the top? Can't quite tell.
It's a little more bulged out, I think you're right.
Oh,
I think it's almost like it's collapsed in on the side and squished.
Huh.
Yeah, that's kind of a bummer. Like, we do have aspiring taxonomists, but like they're not being hired.
Yeah, the couple, all the young taxonomists I know are also like cross-trained as ecologists and ending up having to do a lot more ecology type work sometimes because it's easier to get funding for it than just species description.
Thank you for sharing that. Yeah, I saw a post of a scientist trying to name a sponge after SpongeBob SquarePants once. Didn't know you could do that.
Yeah, there's some pretty silly species names out there.
Hey Jaden, I'm gonna come to the south of you now. We got an uphill on the other side of you. So
And we can
flip
look at this
around.
transition where we get moved from the pavement into the sediment. We pick up the bed form ripples. And with the sediment comes some sea cucumbers.
Was it Umbellula sea pen?
Oh yeah, they're fun. Speaking of fun scientific names, Umbellula is just fun to say. So we got a little ways still. We're kind of at the lowest point of the saddle, and we're going to start uphill again, but it's going to be a little while before we get into steeper terrain again, probably, unless there's little local stuff that doesn't show up on the shipboard bathymetry.
I think you can bring your head to the south now.
Okay.
You're gonna.
Can we zoom sponge? If
You're gonna have to come up, Jaden.
Copy.
Or I can feel the rumble.
I was out on deck this morning, but before watch, before the vehicle went in the water, and we passed a squall line. We didn't quite get hit by it. We stayed just south of it for the most part, but you know, it got cold. Like, I'm always amazed at how much cold air can come down in front of those things. Another sea cucumber here on the rocks.
Seems like the rock texture changed.
Yeah, now we're
Yeah.
definitely into the botryoidal, like heavily manganese encrusted. Probably basalt, small pillows or something like that under here. Oh wow. When the wind went from 11 knots to currently peaking at 30.
Yeah, I should automatically go there. Especially while I'm trying to sleep.
Another little glass sponge, another sea cucumber. Oh yeah,
Yeah.
that makes it. It is really hard to do turning point of the A-frame.
It's. I don't know why we don't do that more as a kind of a standard.
All right, I just got an advisory from the bridge to stay inside the control van until the storm passes, if you can help it.
Oh, wow. That bad, huh?
Where?
Yeah.
I can't
Amber,
see
can
that.
you throw up a deck cam? somewhere?
Amber's not. Oh yeah.
No. Sorry, Taya.
Taya.
I was wondering if
could
you could
put
put
it
a
up
deck
here
cam
and-
up somewhere we can see. The wire cam doesn't look bad at all.
One of Atalanta scams.
Can you guys repeat, and I'll try and get it up somewhere.
Oh, we're curious what the... Another little Walteria sponge.
Yeah, Roger. I don't think we'll notice down here at 1,500 meters. Yeah. I got it. I got it. We want to see, what do we want to, pick a camera in there. Cam starboard, cam mid, cam something. Just press buttons. Try, which one do you have control of over there? Oh, yeah.
Yeah, it never looks as dramatic though on camera, no matter what. And it's always a source of real frustration of being at sea, and you're trying, especially in heavy weather, and you're trying to convey how bad it is, and you always look at the video you shot and be like, this doesn't look nearly as bad as it was. I remember once I was out in 30-foot seas, and I was really desperately trying to get a video of it to convey what it felt like to be in 30-foot seas, and nothing did it justice. Yeah, sometimes they don't. Yeah.
There's some good video of Dr. Delaney on the bow of the Thompson, 50 knot winds. And he's got his classic turtleneck on, and poor videographer out there taking video of him. This is what oceanography's really like.
Yeah.
We're all doing it was ripping.
All right, we can start moving again if you're ready.
Yeah, I think I'm ready.
Bridge nav. Let's do 30155.
I think the only time I ever got nervous on a ship in weather, I was relatively new and I was driving the ship, and it was a dark night, and it was really nice weather, and we hit a storm, and the wind jumped up to about 75 miles an hour, and the seas came up right behind it, but you couldn't see where they were coming from. And so we went from really calm seas to the ship running into probably, I don't know, 15-foot plus seas in the course of about 30 minutes, and hurricane force winds. And we had to bring the ship around in that and change our heading, which was a little, so we took 1 or 2 sets on the beam as we came around. Little good-sized urchin out of an old sponger coral stalk, probably coral stalk.
We had on a small dive boat with a 4-point anchor spread in the Hudson River in the middle of December, I guess, late in the year, and it's beautiful weather, sun shining, and we were watching the local.
This
Look at
place.
that.
Watching the local news in the morning, and they were forecasting some
Big sea storm.
crazy seas. We're like, what? The local divers, the local guys that were there. Oh yeah, it's gonna come up fast. It did. Almost sank the boat. Had to cut anchor lines. It was bad. Okay, you can push in there.
Slime star?
Could be. Oh,
Photobomb by fish.
hello. Tried to get that on still. Hopefully it showed up.
Ha! You got the fish. Nice one.
Can we focus just a touch and on the star?
Oh man, I wish that fish was in focus. Ooh. Bumpy.
Very pretty.
What is it?
I think it's a slime star of some type. All right, thank you very much.
Okay, you can go away.
So why are they called slime stars?
Because they make a lot of slime
Where does it come out from?
everywhere.
Oh.
They're the hagfish of the echinoderms.
I think on top of that rock there's a Yoda purpurata.
Really? Oh, yeah.
Yep.
I totally missed that.
I'm not sure if it's the first one we've seen, but
I
Push
missed
it
those
there.
too.
I have no idea what Harold's looking at.
Purple thing right here.
Oh, the worm.
Yoda worm.
so this is a type of acorn worm. Phylum Hemichordata.
Sorry. Come on, Herc. Might be able to get a shot there. You can push right in.
It is aptly named
A little purple.
Purple Yoda.
That falls in, is it? Not a very good angle.
That's okay.
So it's a deposit feeder.
Yep.
You can see it's producing this big, long, sandy trail of poop.
All right. Thank you very much.
Thank you.
How long does their is their tail? Oh, there's the tail. That's a good shot of it there.
Looks pretty long. I think probably where that digestive tract ends there.
Yeah. Okay, off we go.
Yeah, it's a whole nother phylum of animals.
Couple more sea cucumbers here. Interesting seeing this cluster of deposit feeders on the mainly rocky substrate.
You can come up a little bit for us.
I wonder if the surface deposit feeders have an advantage in these environments where it's a hard ground with a light dusting of sediment because they aren't competing for the fresh detritus with the infauna.
That's what actually makes them make a lot of sense. Yeah.
Yeah, it's always surprising to see a bunch of cucumbers on basically hard bottom with so little sediment, but yeah, I don't know. I'll have to ask a cucumber expert in our lab. Yeah, there's research coming out. Some of Lee Miller's PhD work, one of Jeff's grad students, that different sea cucumbers target different types of fresh sediment, I guess. So the mobile sea cucumbers that can float up into the water and swim might be going for the more fresh stuff.
Well, be good for a move there, Chris.
Yep.
Flattened out here a bit.
Bridge nav, 30150.
Another Metallogorgia there, up on the left.
Sorry, what'd you say?
No, I just commented there was Metallogorgia. We don't need to stop and look at it.
Right.
They're one of the few things that down here that really can't be anything else.
And Metallogorgia melanotrichus, I think, is the only species, right?
It's not a, I don't remember if there's one or two. It's certainly not a very speciose genus.
Yeah. I had kind of thought that it was
There
a
might just be one.
monospecific genus. I'm now going to double check myself.
I missed, I don't know if you saw it. I missed.
Cryptic species aside,
Where is it? Where is it?
because you never know.
I missed that guy.
What, the coral and the sponge?
Yeah.
I'm pretty sure it's one of the Tritopoloria we've been seeing.
That's the hill we just came up.
Yep.
Sorry, making you dizzy there. I wasn't sure what it was. I saw it in the aft cam.
So there's four?
Looks like 4 species.
Yeah.
At least when I've annotated Pacific video, at least according to Megan, the vast majority of them are melanotrichus.
Thank you for tuning in on nautiluslive.org, joining us on this 23-hour dive on unnamed seamount. How's everybody feeling this morning?
I'm ready for breakfast.
How cold are you?
I'm getting pretty cold, not gonna lie.
Kin, how about you, Kinlan?
Very, very, very cold.
Like 1 out of 10.
Colder than my freezer at home.
How about front row? Is it pretty cold up there?
No, it's like, it's totally balmy up here.
It's pretty warm, yeah.
Cold in this corner.
Cold in
Chris
mine,
is all
yeah.
frozen.
So if you're a frequent viewer of the stream, you probably hear us complaining about how cold it is in the control van quite a bit, and that's because, well, A, some of us sit right next to the air conditioning, and B, the air conditioning has to be pretty low because of all the computers in here, and we obviously don't want the computers to
Look
overheat,
up
so
just a bit.
they take precedent over our poor little warm-blooded human bodies.
I do think we could get some ducting to run the air conditioning from behind the back row over to the racks, which are perpendicular
Yeah,
across
where
the
the-
control van.
Yeah,
Oh,
Yeah,
it's-
it is.
where the
It is
Trust
kind
me.
of
coldness
funny.
needs to be.
It's
The
been
cooling
discussed.
system for the computers has to go through us.
It's not necessarily the computers, it's the television broadcasting station over there in the corner.
Ah,
Which is made up of computers. Can we look down the axis of the...
Yeah, we can.
Is it made of computers? I think it's a little more analog than that.
Yeah,
Yeah, it's got wires and lights and buttons.
more
Yes,
digital.
exactly.
It's a computer.
There's miles of fiber optic in there and some really high-powered video hardware.
Yeah, which are all basically computers.
Yeah,
Whether they work on firmware or software is immaterial to us biologists.
you go ahead and
It's all electrons.
push it in.
They work on frequency oscillators and things like that, I think, not digital.
What is this, an analog station? It's all digital.
It is digital.
Is it?
Yeah, 3G.
We've got a
You
glass
could go
sponge here with a...
full zoom on that.
Symbiotic shrimp living in it.
So we have another sponge dungeon.
Sponge dungeon.
Dungeon. Yeah, so this shrimp
heard that
actually
one
has no
before.
way out of the sponge. It lives there its entire life.
What's the little bug on the outside there?
Yeah, that looks like a amphipod. I love the little bright red
Yeah, so tiny too.
face of it.
All right. Science sappy, thank you.
Moving on.
All right. Good to keep moving?
Yeah, so for the viewers who tuned in, this will be our
Not
last
sure
dive
where
for
it is.
this
Yeah,
Eagle
need
Voyage
to
2.
another
Bridge nav,
Hop
30
around
meters,
quite a bit.
150.
And I could be there, or I could be there.
Yeah, it's impossible to say. It looks like you're pretty on top from the camera view.
Yeah.
I don't know, it's stabilizing there.
What's that?
It's stabilizing on Sonodyne.
Given Jaden's
Hang on,
camera,
this is what's,
given
somehow you're,
Jaden's camera angle, I would say that
that was the issue.
that's
Somehow
where
we got, I just reset it. I don't know what the deal is.
they were both hopping around.
Yeah.
So if that shrimp lives there its whole life, how does it reproduce? Does it reproduce? Will it?
Yeah, a lot of times you see a male and female pair where they live there together, so they can lay eggs, reproduce, and their larvae can leave, and they settle and go find their own sponge to settle in.
Oh.
The sponge provides them protection, and they can feed off of the leftovers from the sponge, basically pulling the water through. So sponges actively filter water, and that they have these choanocyte cells or flame cells that actually force water to move through them, whereas corals are passive and have to basically just wait for the current to bring them food.
It's really cool to pump fluorescein around the base and watch them waft it out.
Yep, totally. And then
Exhale through the
the different
oscillate.
growth morphologies of sponges are optimized for different current regimes.
See, inhale or exhale through the
Exhale.
oxygen? Yeah, exhale. Been a while.
Can we look pale yellow coral,
You want a quick zoom there?
please?
You can push in about halfway for me.
Look how strong the current is, just vibrating in the wind.
Yeah, I can feel it. Okay, you can push it a bit more. Hercules weather vanes around here into the breeze.
So Dan, when you said you can feel it, does that mean like it's harder to maneuver Hercules, or what's happening?
We're good, Dan, thanks.
Roger. Okay, you can go wide. I wouldn't say it's hard, it just when we touch down Hercules on the front of its skids there, then it naturally weather vanes around into the current.
Yeah. The DVL keeps jumping. Teleport.
Teleported. It's respawning.
Great. Love to see it.
Need more beams.
Need more something. I don't think it's the DVL. I think it's the...
Ship going sideways.
No, I think it's the software not rejecting outliers properly or something. Or not at all.
I guess you kind of get a feel for it when you're trying to move Hercules one way or the other, so. And After a while, it becomes kind of second nature, like if you're riding your
Yeah, look at
bicycle.
it. See how it's shaking off? DVL is being weird.
Yeah, it is. In a crosswind, you naturally, or a motorcycle or a car, you kind of, you know, steer into it.
Yeah. I'm going to switch us to USBL for a bit,
Or
considering that appears to be whack.
Is that the technical term?
That's correct.
go back to the 10 meter squares too, that'd be great.
Yeah.
Thank you.
I left the snail trail on so we can see what it's doing. Yes, sir. All right, I'll hold this position, I guess, until the watch change. We got, do we have, is something going wrong with Sonodyne? This is like, I'm not happy.
Where are we? I don't know.
Why are, oh God.
Chris, I don't think that's right.
Well,
And can I put the normal layout back up?
We
So
have,
that
oh.
Yeah.
Huh.
Oh.
So it's saying that we have bad GPS fixes on the ship. Yeah. Poor GPS quality. No, it doesn't actually tell me anywhere what the GPS quality is. It just tells me that it's bad.
well, luckily the vehicles are attached to us, so we can't lose them even with bad nav.
I wouldn't go that far. Wait. What is... I don't, like... This looks different than this. Doesn't it? How... no, maybe not.
Let me,
No, that says it's 80
see.
meters away.
Jaden, if you bring your head to the left just a little, maybe south, we'll look up the hill here. We'll get our ducks in a row for watch change here.
All right, for our viewers,
That's good.
we will be going through a watch change. There's a 48 shift. We'll be back in 8 hours. So hope you have a good day. Continue watching
Yeah.
L5.org.
We're gonna sign off here in the front row and discuss our where we might be, somewhere in the South Pacific, Philippine Sea, we think. Thanks, everyone.
Hi.
Sonodyne issues.
Nope.
No, just pretend the ship is closer to the ROVs. Just pretend the ship is... Oh, really? I'm on SBL. Yep, I'm talking SBL. I can talk, and I'm talking directly to you as well. Oh, well, yeah, Dan, sometimes with the dual headphones, you don't have to have the volume up so high. Yeah, I think our configuration is fine. It's just the ship is not in the right spot. So just use your imagination. That's USBL anyway. That's pretty much sums up USBL. Use your imagination. It's just where we are.
Yeah. Sorry, guys. I'm getting settled. But yes.
No worries.
Yeah, just give me 1 second here.
I am so settled.
Super chill.
What? Is there a tiny fish in there? That's probably a shrimp. Yeah.
Brittle star. Yep.
Hello.
Okay.
All right, go wide.
Jeff, are you ready to start heading up slope?
Yeah, let's start heading up slope.
Okay.
Okay.
Getting the last of the things set up
Yeah,
here
those
on my
night
side.
creatures look like they're, what, like 50-ish meters away, so...
Yeah, let's step slowly into the hill when we get there.
Yeah.
Yep. Yeah, looks like we've got a little flap first for... Oh. We've got almost 500 meters of pretty mellow slope before it gets steep.
Nice.
Could
Well,
be
I'm
fish
seeing
territory.
something 60 meters away in the sonar. So,
Oh,
that'll
okay.
let the sonar,
Yeah.
so that's
Yeah, there's a boulder. Looks like.
pretty good shadow behind it, so that's definitely something.
Yeah. Okay.
Good morning, everyone. I'm super excited considering this is our last dive of this particular expedition. Whenever you're ready and settled, could we do introductions? Are we good?
Sure.
Awesome. So I'll start things off. I'm Amanda Dedicatoria, Ali, and good morning. I'm a freelance writer and filmmaker from Guam, and I'm joining you as your science communication fellow. I guess today we'll do the back row. Menzel?
Good morning, everyone. Ali, my name is Menzal Medil. I work for the Office of the President here in the Republic of Palau, and on board I'll be the cultural liaison for you guys.
Right.
Good morning.
Thank you.
Ali, everyone. My name is Jeff Drazen. I'm a professor in the oceanography department at the University of Hawaii, and this watch's science lead. And I'm a deep sea ecologist, and I specialize in deep sea food webs and in deep sea fishes.
Unil Tutao, everyone. My name is Greta Sartori. I work as a researcher for the Palau International Coral Reef Center. Today I'm going to be sitting in science seat number 2 and give a hand to Jeff, take some still pictures, and also in this moment I'm actually in the data logger position until our data logger person is joining us soon.
Two jobs.
Like your two hats from yesterday that you wore,
Yeah,
one on top of the other.
Were you wearing two
Now
hats?
you know why.
You're amazing. Thank you for your hard work. Oh, let's go with Dan this morning.
Sure, good morning. My name is Dan Torriello. I'm a research project manager at the Center for Coastal and Ocean Mapping at the University of New Hampshire. And today I'm serving as your navigator.
Thank you so much.
And hello, I'm Rye. I'm in the Herc seat for the start of this dive.
Hi everybody, Josh Chernov, ROV Operations Manager for OET, and sitting in the Atalanta seat today.
Ooh, exciting. Love a switch up. And last, but definitely not least.
What is this?